until 9:44. All right, so we're talking about cell membrane. All right, and we already mentioned this. I think I mentioned this before. It's the cell boundary. It's the boundary between what's inside the cell and what's outside the cell. And we say that it is semi-permeable, so some things can get through and some things can't. Permeable. So basically it's semi or selectively permeable. It lets some things through and some things not. What is it made of? It's made of these things called phospholipids. So made of phospholipids. This is a phosphate molecule on the top, and it has a bit of a negative charge. All right, it's polar, I should say. Let me change that and just say that this thing is polar, and that comes back from chapter 2. That idea that water's polar. So that phosphate head on this molecule is polar. Now there's two lipid tails. Lipids are fats and oils, remember? So I'll just say fatty. So they're like oily, fatty or oily tails. Now here's the thing. The phosphate is, here's another term, hydrophilic. Is there two L's on there? Yes. No? Just one L. Hydrophilic. Hydro means water. And this comes from phile, which means love. Water lover. So it likes water. It's attracted to water, okay? The lipid tail is hydrophobic. And that means water fearing. So what happens when you put a bunch of these in water? If you just had a bunch of phospholipid in a bottle, and that's possible. If I had a bunch of phospholipid in a bottle and I poured it into water, it would naturally do this. It would form little things like this, where all of the tails would go toward each other, because out here would be the water. And so what would happen is, if you got a bunch of water out here, the tails are afraid of the water. They're actually, what's really going on here, it's not that they're actually afraid, right? They're not people. They're not afraid. Water has such a strong attraction to itself that it, because it's polar, that it excludes things that it's not attracted to. Have you ever been excluded from a group because that group was so tight-knit, there just wasn't room for anybody else? Right? That's what we're talking about here. Water is so tight-knit to itself that you can't get in there unless you also have a charge or you're polar. You can't mix with water unless you have a charge or you're polar. And so if you're in oil, you don't mix with water. You take your salad dressing bottle, you shake it up, it mixes real quick, and then what happens? It separates out again. What's happening? The water is squeezing the oil out of itself and pushing it away. It only goes up. It goes up because the oil's less dense than water. So this is what would actually happen. This picture right here would happen if you just poured some phospholipid in a bottle. And actually, you know what? Soap works the same way. Soap has a polar part and then an oily tail part. And so soap can mix with water and oil. That's why soap can get oil off of things. because it can mix with both and it forms like a link between the two things. So picture a soap molecule. Let's say I have a big glob of oil here. What the soap would do is like this. So this would be soap on this side. It would do this, and then what happens is it can link up with the water, and now you can wash the oil off with water because the soap forms like a link between the oil and the water. It surrounds the oil droplets. Did you ever get butter on your hands and try to wash it off with just water? It's not working. No. But put some soap, come right off. So that's how it works. Now, in the cell membrane, what we have is a bilayer, where somehow they get organized like this. And I don't know exactly how that happens, but they get organized with tail to tail in a layer like this. All right? Now, water can't pass right through it because water would have to go through the hydrophobic tails, and it can't do that. It can't mix with the tails. So what we have are these channels made of proteins. Okay? This is a protein channel that things can go through, right? Like water. So water can go through the membrane. All right? Water can pass freely through the membrane whenever it wants. But not all things can. So let's write this down. Water can pass freely through the membrane, but salt and sugar can't, among other things. So I'm just saying salt and sugar plus lots of other stuff. That's for example. Can't. Now is where this whole thing about osmosis comes in. All right. If I have a cell, and I'm going to draw the double membrane. I'm going to say I've got two cells. All right. That's a bilayer, yeah, because it's two layers of phospholipids. So here's the membrane. These are two cells. Now inside the cell is water plus dissolved stuff. Ions, glucose, etc. So it's the same thing, water plus ions, glucose, etc. All right? And remember there are channels in the membrane so that water can get out. Okay? Now, here's the thing. Let's say outside of this, we just had a bunch of water, and it was pure water. Let's say that outside the cell was pure water. Well, there's this principle called diffusion. Okay? Diffusion is how molecules spread out through substances randomly. Molecules spread out randomly. All right? Or the way that your book says it, they move from where they have a high concentration, where there's a lot of them, to where they have a low concentration. They move from a high concentration to a low concentration. So here's the thing. Is there more water? Like if I said the concentration of water is greater where? In this picture, the concentration of water is greater out here because this is pure water. In a sense, the water is like diluted by, the water's not diluted, but it's a lower concentration inside the cell because there's other stuff in the cell. It's not pure water. So what will happen is, I'm almost done. What will happen is the water will move into the cell in this picture. because the water is more concentrated outside the cell than inside the cell. And the ions can't come out. So the ions and stuff can't come out to make the water less concentrated outside. That can't happen. It's only one way. Only one thing can pass through the membrane, I mean, in this case, in this picture. Now, what if I have salt water out here, and this is what we're going to do in the lab. Now, the water is more concentrated in the cell than outside, so water will pass out of the cell. Water will come out of the cell because it's moving from where it has a high concentration to where it has a lower concentration. In other words, there's more water per, say, milliliter, more water molecules per milliliter outside than there are, well, in this case, there's more outside than there are inside. In this case, it's the other way around. Now, what we say in this situation here is that we call this solution hypotonic, and then I'll be done, and this one hypertonic. Now, in a hypotonic solution, the water has the solution outside is less concentrated with the stuff that's dissolved in it, and then in a hypertonic, it's more concentrated. So like salt water, or those of you who are doing the egg lab, you've got the egg soaking in corn syrup, that's hypertonic.
Wait, what does the salt water do?
The salt water is what's going to do is essentially it's going to dry the cell out. Because water is going to come outside of the cell. It's kind of like the water is trying to dilute the salt water. Another way you can look at this diffusion in osmosis, another way to look at it is the concentrations try to equal out. What is concentration, by the way? I guess I should define that. Concentration is the amount of dissolved substance, or the amount of substance per volume, I should say. Amount of substance that you're talking about per some volume. Like per liter, for example. A concentration would be like saying there's this many grams dissolved for every liter of the solution. All right. So I said I would do